Hey everyone, it's Mr. Wolfe here, and in this video I'm going to be discussing, or at least going through the thinking of what you guys could be doing when it comes to forming your explanations for the evidence that we got in our liver lab. So just really quickly, to give a brief overview of what we did in the liver lab, is we had these test tubes filled with a substance called hydrogen peroxide, also known as H2O2, and then we added liver into the test tubes. Now the liver had this enzyme called catalase, and the function of catalase is to break down the hydrogen peroxide. So when we did add the liver into our solution, it produced a bunch of bubbles. And then what we decided to do is we tested different types of scenarios and to see if these scenarios would produce more bubbles, less bubbles, or even no bubbles at all in different circumstances. And it's our goal from that evidence to be able to explain why were there a lot of bubbles produced, why were maybe no bubbles produced. Okay, if you were absent, really quickly, this was the data we ended up getting. So for the first experiment, we just changed the amount of hydrogen peroxide we had in each tube, and when we only had 1 milliliter of hydrogen peroxide, we had a low bubble level. When we had 5 milliliters of hydrogen peroxide, we had a high bubble level. All right, for experiment 2, I don't have the actual page with me, but we had a raw liver, so this was similar to experiment 1, and this is the same as liver that's at room temperature. But then we also had a boiled liver, and the way we could think of this is this was just liver that was at a really high temperature. And when we had liver at room temperature, we had a high bubble level. When we had when we had liver at a room temperature, we had a high bubble level, and when we had liver at a high temperature, we had either no bubbles or a very low amount of bubbles in our test tube. All right, finally, last experiment is that we had very similar to experiment 1 and 2. You just add raw liver to the test tube, and as we've been seeing, you get a high amount of bubbles. And then what we did is we took the liver out of that test tube and we just put it in an entirely new solution of hydrogen peroxide, and we saw the same thing, a high bubble level. All right, so that was our data. So our goal is to be able to explain this evidence, and in order to do that, the first thing we need to do is just state our evidence. So I'm only going to be going over experiment 1 right here, and we've been providing evidence all year long, so you guys should be experts at this. And remember, anytime I write evidence, I want to have my specifics and my patterns. So this is what I have, is that liver placed in a test tube of 5 milliliters of hydrogen peroxide produced significantly more bubbles compared to liver placed in 1 milliliter of hydrogen peroxide. And notice that I said hydrogen peroxide here, spelled it out, and then I used the formula for it there. You could do either one. All right, so in terms of my patterns, the big thing for my pattern is that I used the word more, and then my specifics was that I talked about my actual solution, hydrogen peroxide, and then I said what amount was in each trial as well. All right, so there's my evidence. Now, the next thing I need to do is I need to construct my explanation. That's my ultimate goal, but it's going to be really, really helpful to draw a model out in this case. And by drawing that model, it hopefully will make a little bit more sense in terms of why we see a low amount of bubbles here versus a high amount of bubbles here. So one thing that I'm giving you guys is these boxes on each type of lab, and the answers to these boxes will help you a lot with modeling what's going on, and then finally answering your explanation too, or making your explanation. So this box says is how could you model 1 milliliter of hydrogen peroxide versus 5 milliliters of hydrogen peroxide. So what I'm going to do is I'm going to say, okay, this is going to be 1 milliliter, 5 milliliters. If you go to the Google slides, any of the slides that say liver lab on them, it should only be about 4 or 5 of them. One of those slides tells you how we're going to model hydrogen peroxide. And we model hydrogen peroxide just with like a big circle and a smaller circle. So in this case, if I have 1 milliliter of hydrogen peroxide, in reality we have millions and billions of these molecules, but for my purpose, I'm just going to draw 1 hydrogen peroxide to represent 1 milliliter of it. And then for 5 milliliters of hydrogen peroxide, I'm going to draw 5 molecules of it. Okay. So then it goes down. Then we go down to our model, and it says draw draw a model for 1 milliliter of hydrogen peroxide in terms of what was happening in my reaction. And so if I had 1 milliliter of hydrogen peroxide, I'm going to draw 1 milliliter of hydrogen peroxide. Now I also know that in my test tube I had liver. And specifically, in the liver, what we care about is the enzyme called catalase. And I know that if it's acting on my hydrogen peroxide, it's going to have that active site where that hydrogen peroxide could bind perfectly to it, and then it could split it apart. All right, so once this reaction occurs, I know that enzymes could be reused again and again, so the shape of the enzyme is actually going to stay the exact same. At least it's intended to look like that. And then it's going to split up the hydrogen peroxide into oxygen and water. So this is my oxygen, and this is my water. And if you're not sure which one is which, if you go to the Google Slides, it'll tell you how to model oxygen and water in this case. I'm giving you that information. It's how you use those tools to figure out what's going on here. All right, so now I'm going to draw a model for 5 milliliters of hydrogen peroxide. And I already know how I'm going to model that. I'm just going to draw more hydrogen peroxide molecules. 5 in this case. And I'm also going to draw my enzyme. Now keep in mind the liver has millions of these enzymes in it, but for our purposes we're just going to draw one enzyme. Remember, enzymes don't change before and afterwards, at the very beginning, at the very end. And so I'm going to draw the same exact thing, but once this reaction takes place, hydrogen peroxide is split up into oxygen and water. However, if I have more of these hydrogen peroxide molecules, I'm going to have more oxygen and more water as well. Okay. So what I want to do now is I want to look at my evidence. I want to see if my model could explain what was happening here. So for the 1 milliliter of water, I had a low bubble level, and the first thing I might want to wonder is, well, what is the bubbles in this case? And it's going to be kind of similar to our photosynthesis lab that we did, is that the oxygen in this case is going to be those bubbles. So I'm going to start to write that in my explanation. I'm going to say that the bubbles are the oxygen gas that is produced by the enzyme catalase, speeding up the breakdown of hydrogen peroxide. All right, so at least I've established what those bubbles are. And then I still need to show or explain why did we see more bubbles with 5 milliliters of hydrogen peroxide. Well, if I just look at my model right here, is that, well, if I have more hydrogen peroxide, more of it's going to get broken up by the enzyme catalase. Remember, enzymes can be used again and again and again. So in this case, I just had more hydrogen peroxide to be broken apart, and I'm going to get more oxygen. So to kind of put that in just one nice sentence, I said, because there's more substrate in the 5 milliliter tube, catalase was able to help speed up the creation of more oxygen bubbles. That's it.